We were interested in addiction and depression, and we had some tissue that was given to us from a researcher at the OHSU. We were interested in kind of some sex differences that we see in addiction and depression. Females are twice as likely to experience clinical depression as males. Males are more likely to become addicted or have addictive symptoms than females.
Maybe there's circuits in the brain that are developing in the womb that predispose women to have a higher risk of depression than males. So we used sheep as an animal model, and we looked at serotonin cells in the developing brain.
For depression, we looked at the number of cells that were expressing an enzyme called TPH2, which stands for tryptophan hydroxylase 2, which is the rate-limiting enzyme in the synthesis of serotonin. We wanted to look at the levels of serotonin in the brain to see if differing testosterone levels resulted in changes in serotonin expression or presence. And then for the addiction study, we looked at the neurotransmitter dopamine.
Males will perform much riskier behaviors to get illicit drugs. And so we thought that we'll look at the dopamine system too, which is involved in reward. What we found was in the reward circuit, sure enough, consistent with our hypothesis, males have fewer dopamine cells in this circuit. There's many reasons why people become addicted to substances. Here we have, at least in an animal model, a suggestion, maybe one little piece of the pie that would suggest that there are differences here that might predispose a person to addictive behaviors.
Both addiction and depression are two diseases that affect the human population. If we can kind of disentangle what we think might be going on in the pathology of both diseases, there's potential to help a lot of people.
Here in the genetics lab, we obviously work with DNA. My project is trying to analyze an ancient Roman population excavated from the city of Palencia on the island of Majorca, Spain. Dr. AR, myself, and 3 other students traveled to Palencia to participate in the archaeological excavation ourselves. We were working in the field for 5 to 6 hours a day, and then washing the bones and pottery that we found for another couple of hours.
Typically, from Christian background, we're buried on our back with our hands crossed. They also came across skeletons that were born in more of the Islamic style, where you're on your right side facing Mecca.
So we're trying to look at this population and use DNA to answer the question, where did these people come from? We start off with the human bone, and we don't have them yet. They're in the process of being exported. So this is just a sample. But once we do get the bones, what we'll do first is a very thorough decontamination procedure, and we'll drill into the diaphysis or shaft of a long bone to obtain some bone dust for the sample. Once we have that little bone powder, we will perform DNA extraction and isolation on it using a kit made specifically for ancient DNA. So that'll give us the DNA in solution by itself. And once we do that, then we need to amplify the DNA that we want to look at. And at that point, then we can send the sample off for sequencing to an external company, and then they will send us back sequencing chromatogram data, which we can read and actually get the sequence of DNA itself. Then we can assign kind of ancestral origin to these individuals.
Some of the work we do will help contribute to databases worldwide, so other researchers will be able to look at it and answer some of their own questions.
You don't want to have a lot of high levels of metals or nutrients in water runoff, just because when it gets to its destination, that could be dangerous for fish or anyone affected directly by that body of water.
Joselle helped me take samples over a 1-year period to look at the water quality and how it changes over time.
A lot of people look at eco-roofs for temperature retention, but there isn't too much research on stormwater quality from eco-roofs. So anytime there was a rain event, we would go out to the drains that are down on the lower level on the outside of Shiley. And there were 2 drains we collected from, a drain that came from the eco roof and a drain that just came from the regular roof. Once we had enough samples, we would start running tests. We would make concentrations for zinc and copper, and then we would use our atomic absorption spectrophotometer to test the metals.
We've realized over time that because we use compost in most of these systems, we have phosphorus that leaches out of the system. And so in the last few years, researchers have collected water quality samples and realized that we have a large amount of phosphorus that's leaching out of these systems. Once phosphorus gets into the river, there's all sorts of water quality issues that's not good.
There was definitely a correlation. There were definitely higher levels of phosphorus at the beginning, and you could kind of see throughout the months that it kind of dissipated.
So I really want to try to help be part of fixing that problem. We make some progress and then realize we need to do something better, and we just keep moving forward.
One way we could prevent having high levels of phosphorus in our stormwater runoff would be by choosing a different kind of fertilizer or soil for our eco-roofs.
We have had the opportunity to kind of be a part of the whole scientific process.
They do pick up tolerance, they do pick up appreciating where data comes from.
Fundamentally, that's what research is. It's trying to figure out what we can do better and answer questions that we have.
That initial time that you have found something out that nobody else knows, only you. I mean, it's very exciting.
It was very eye-opening, and it was great to get some hands-on experience just because I had never done something like this before.
They learn all of the analytical techniques and the standard methods for sampling. And so if they decide to go to graduate school, they're already trained.
I'm certainly proud of them.